Hello.
Hi, James. How are you? Thanks for coming.
I'm great. Thank you. Thank you for having me. Uh, just just working on setting up the microphone correctly so you guys can hear me better.
Okay, yeah, I'll in the meantime share on Twitter and so that we are about to start. Um, hey everyone, feel free to share the room with people you think that might be interested. And I'm doing the same. I'll be unmuting in 30 seconds. Okay. People will start joining, but in the meantime, as I said, I'm sharing also on Instagram that we are starting. Yeah, it's things you can only do acutely when people are here, because if not, it doesn't make any sense to share like a link where I'm just mentioned, like that I'm talking with myself.
Well, I
How's
hope
your
it's gonna
day?
be. Yeah.
Oh, for you it's already evening, right? So how is
Yeah, it's 7 p.m. here, but it's kind of my morning because I am rather nocturnal. I woke up like 2 hours ago, so it's kind of my morning too.
Okay, that's perfect
Yeah.
then. I'm
Yeah.
usually also a night owl, so I totally get it.
Yeah.
And it's wonderful that you found, a way to adapt the life to this.
Yeah, yeah. It was really crucial because I was like this for all my life, and I couldn't have survived, I think, because when I was younger, I think it was easier, but I think I'm getting a bit older now, and it would be so hard to like I have a schedule from 9 to 5. So I'm really glad that I can just do things in my pace.
That's perfect. I'm glad to hear that. And I think we can slowly start by
Mhm.
telling the audience who you are and so on. And then, as I said, in the meantime, people will start dropping in, but I think they
Mhm.
will be able to catch up um, pretty well. So, welcome everyone to Science Society, and of course, a special welcome to James.
Yay.
And I was so excited about this room. Don't let other guest speakers hear, because James is an independent researcher doing his microscope work from home. That's my dream since. And James is also a really famous YouTuber. He published papers and a book, and he collaborates with academia and also other people around his YouTube channel and their store even. And so yeah, check it out. I've posted the resources in the chat, and on top you can see this really cool YouTube channel that I've been watching recently quite a lot. So I'm so glad I found you and that you agreed to come here. And we kind of thought that it would be the most interesting way to make this kind of a like informal
Mhm.
discussion, because I think there's so many interesting aspects your life and then also your work. So I
Mhm.
think that's the best way to go. So the first question I have for you is, how did you find your passion? Because, when we talk, like, to prepare a little bit, you said,
Mhm.
you managed a way to, to turn your passion into, the life and work you do. So was that
Yeah.
something you always had, like, since childhood? Was it maybe something that triggered it, a book
Mhm.
or
Mhm.
a class or something? How did you find your passion?
Well, it was with a lot of tries, um, but I think since I was a child, I was fascinated by animals. It was it was so amazing to see I don't know, documentaries and like photos on these like old magazines like Nation Geographic and stuff. And I don't know when it started exactly, but I was always crazy about animals. And when I was a child, I wanted to be an author and work, with animals, just live in Africa, somewhere in Africa, just do observations and stuff. But um, well, when you grow up, you face the reality. I think it's a bit um, it's different than these innocent childhood dreams. So and then I ended up in Poland somehow, and I was studying engineering here. And in one of our lab courses, we needed to look through a microscope and just observe these things that were living in the wastewater treatment plant. And so cities uses uh, s- um, one more like wastewater treatment is done with the help of microorganisms, and this was one of the aspects of the lab course. And then I just looked through the microscope, and it was just, love at first sight. It was unbelievable. I've never seen anything like that. It was just unbelievable. There were like so many things moving, swimming, and like I am crazy about animals, but apparently there was a whole universe that I never thought of before. And just seeing that in front of me, it was unbelievable. And that was, I think, 6 years ago, and I cannot do anything else. But I cannot think anything else than microbes. Everything I do is just about microbes. And what I did, so the first thing, the lab course was 2 hours per week, and it wasn't enough for me. So I needed to get a microscope to just start doing this, in my living room. So I got a really cheap microscope, and then I started using it, and I learned how to use it, and I started making videos. And well, it led to this, and I'm here now talking about my life to you, to the audience, and it just becomes something. And I didn't anticipate that. Like I didn't expect that. It was just I don't know, just happened.
Well, I think that's wonderful, because, when we have passion for something, then we become really good at it. And I want to point out here how great your images and videos are. Like, the quality of the videos and images that you put on these YouTube channels, they are amazing. And so, how do you, like, how did you train yourself? Basically, I assume that you trained yourself. Did you take courses? Did you completely train yourself to hone these skills that are quite, quite wonderful?
um Well, I don't think there's anyone in the world that can, teach you the microscopy in a way that you would learn just by trying it. So it is, um, it's borderline art and science. It's a very strange thing. So it fits so perfectly to the way of me observing and like observing life and just looking at things. So with the microscope, it's just a tool, and I use the tool to just create aesthetically pleasing stuff. And there are a lot of factors affecting the quality of the footage. So it's not just the equipment, or it's not just the way of you setting up things, but it depends on the person behind the microscope and their eyes to just find these beautiful sceneries and just put them in an angle that would make people just say wow. So it's just combination of art and um, it's it's really hard to quantify it. It's really hard to say that you do this thing and then you get this thing. So I don't know. It's just like a painting. You just get something, you just get an empty canvas in front of you, and then you start painting, and it becomes something. So that's how I feel when I do microscopy. And I took no lessons, and I'm not sure if there are any lessons like courses that teaches you how to make videos through a microscope. We are actually trying to do that because we recently created this um, microscopy brand, and yeah, so we need to teach people how to do that. But we also need to first understand how we can do that, how we can teach people. So it's yeah. Uh, Sorry, my cat is
That's
eating very loudly. I hope
oh,
it's not
it's fine.
it's not bothering.
We no, it's we have children in the back. I for sure you will hear my kids in the background. They're in the backyard. We will have different sounds. It's actually nice, so we kind of feel at home. Um, it's really impressive how you did that, and at the same time, I just wanted to point out it's you combine that with this really aesthetic art of microscopy and video making. You combine that with finding new species. I think you wrote me, you published the paper. The paper for everyone is in the chat, and everyone can just access it. And then you wrote me that you will publish more because you found, I think, over 35 new species. So how do you find them? Like how what you just go to everything you find, or do you have like a strategy? It's really interesting how you find these new species.
Well, what I do is I just collect samples and I check them under the microscope. So that's my strategy to find anything. But it, like, I'm not trying to specifically. I'm not trying to find new things or make discoveries. These are like my paper or these new species or anything else. These are just. Um, can you hear me?
Yes. Yeah,
Ah,
it's
sorry,
perfect.
sorry.
Yeah. Sure.
So yeah, sorry, I got a call. Sorry. Uh, so I. Oh my god, I'm so sorry. My professor is calling me. I don't know why.
Oh, don't
Uh,
worry.
uh, Sorry, I got distracted. And just a second.
Yep.
Uh, yeah, so I don't try to do discoveries. So I don't try to just discover new things, but I just live my passion, and these things just appear. So I do microscopy most of the days, like 12 hours. So I'm sitting in front of my microscope. and checking bunch of water samples from different places, and occasionally from different countries, from different continents. So when you do that, you just come across to new things. And so I specialize on one group of microorganisms called the ciliates. These are microscopic eukaryotes. Like, we are also eukaryotes, but they are just single cell, and they create this very interesting group of you know group that's called the ciliates. So I specialize on these on this group. And I have a like, I don't want to sound super cocky about this, but I have a good knowledge about everything that is in literature about them. So when I do microscopy, when I see something that I don't know, when I see a ciliate, more specifically, that I don't know, there's a good chance that it's going to be something new, or it's going to be something super rare, just described or mentioned like 100 years ago, and nobody has ever seen it since then. So when I see stuff like this, first I try to document it, try to record like good footage of it. And then, depending on the rarity of it, I isolate it. I wash it, I clean it, from all the other contaminations, because I want to use them for. I want to use them to get the genomic data from them. And then I put them in tiny, super cute Eppendorf tubes and just put them in my freezer. And yeah, so that's how I do the discoveries or discover new species. And these 35 new species, these are the ones that um, have or will have super soon the genomic data, because without the genomic data, it's really, really hard to publish something and say, okay, we made a publication and this is something new, but we don't have the genomic data. So we need to have a bit of this, modern um, sequencing of the partial genome of these new things. And then with the publication, that we can publish it, and we can just make it accepted, and yeah, introduce these new things to people.
Yeah, that's interesting. That's so impressive that, whenever you see something that you don't recognize, it's probably not discovered yet. That's so impressive.
Yeah, it's so I call them like my friends, because you have this social group around you. You have, well, if you're super social, you have like 10 people around you, right? You don't forget them. You don't forget them. So when you see them, you know that you know them. So it's like that, because I am just living with these ciliates, and um, just I'm sleeping next to a bunch of books, and so I spend a lot of time, and they are my entourage, and I know every single one of them. So when I see a new face, appearing just in front of me, I'm like, hey, who are you? Like, can we be friends? So it's, yeah.
And I've recently read a a bunch of reviews and articles saying that the microorganism diversity and population is going down, especially in developed countries. Do you think you're working against the clock, or do you think it's a little bit too much? Because, like, a hype or overstatement, because
Mhm.
you look at them probably more
Mhm.
than most people. So
Um, well, I think it's more like fear mongering. I think these kind of articles, they are just trying to get attention by just scaring people. Um, so with microbes, that's a thing with, with mammals or animals, like the diversity is just getting, you know, lower just because of the human activities. But with microbes, that's definitely not the case. And if they're not going to be around the industrial places, but they're just going to be around somewhere else, because microbes, they are cosmopolitan, meaning like every single species, you can find them everywhere, all around the world, because they are around for millions of years. Although they evolve, they change, like everything else, they disperse so easily because they are so small. So if you find their habitat, like for example, if a microbe it likes to live in, like this humid, hot climate, you can find this microbe in Miami or in somewhere in Kenya. So it just depends on the habitat. So as long as that those habitats exist, microbes gonna be all over the world, and they're gonna be everywhere, and they definitely gonna be around even after we are gone. After we just, I don't know, nuke ourselves to oblivion and stuff. They just gonna be around, and yeah.
Yeah, and
Uh,
you have a video about the definition of death related to microorganisms, and that video is really interesting. Would you like to a little bit talk about that? Like, what
which
changed?
one is that?
Um, um, I I don't know the exact title, but
Uh-huh.
what death is, or what death means, or what dying means.
That. Okay. Yes. Yes.
Yeah.
Yeah.
And
Sure.
yeah, did your view change when you started looking at and learning about these different microorganisms and how they kind of some cheat death in kind of a way, and they have this amazing abilities to
Hmm.
reassemble, and how would you define, yeah, death for microorganism compared to, like, a general view of death? I think that was so interesting.
Um. Yeah. So the thing about these microorganisms, when they reproduce, they create they create clones of themselves, right? So the definition of death is really strange, because like, if you exist, how can you be death, right? So there are like many cells dying in my body, but I live. So like in the case of microbes, maybe some individuals are dying, but there are so many other clones, millions, billions of clones, identical copies of that microbe, just swimming somewhere else. So it is it is really strange, and they are rather immortal in that sense. But also there's a thing called like reproductive mortality. So it's a super cool term, I think. It just because when they divide, they stop existing, because one thing becomes two things, and and it's called reproductive mortality. So it just, they die, but they exist as two individuals now. Does that make sense? Am I able to explain it?
Yeah, yeah, it's really interesting. And there was also the mentioned that, about the state of that you can still that of the equilibrium, that you kind of, all organisms kind of fight
Mhm,
against it partially, and that
mhm,
could
mhm,
be a definition, like whenever everything is in
mhm,
equilibrium, including the universe, then that's the
mhm.
ultimate death, which
Oh
is
yeah,
a
yeah.
really interesting view. So that's so interesting that death is something very similar for different types of living things. And would you think what's the which one? That's probably a very complicated question. Which type of microorganism would you think would be able to live like on other planets? Like, what would be your bet if you would have to?
Uh, well, um, it would depend on the planet. But if it's like if there's liquid water, well, there's a chance that every single microbe gonna be finding some places to live, I think. But or you mean if we send some microbes out, and would they be able to just colonize other planets? Is
Yeah,
that the
like would
question?
they survive the yeah, the question is what, however you would like to answer is, but do you think is there one of them that could survive like spreading around the universe more easily on like meteorites or whatever would be, because
Hmm.
I think it's so impressive. Even I have a backyard in the city and there was nothing in the beginning, and now it's full of wild plants that I kind of leave in the corners, and there's
Hmm.
really not a park too close by, but somehow they make their way here. And I don't know, could you imagine that this would be true for the universe too, that there's some sort of system in the macroscopic world to like spread life in
Uh,
interesting ways? And
well, there's one theory called panspermia, which basically, you know, says that life may have come to the Earth from outer space. Uh, well, not life, but the beginning of it. Like I don't know how to define it, but these like the first thing. Um, so maybe it's possible, but we are quite sure that the life originated from within the planet. So, but it doesn't mean it wouldn't be happening, or it doesn't mean it didn't happen somewhere all over the around the universe somewhere. And yeah,
yeah.
okay, yeah, I'm just seeing comments now.
Oh,
Yes,
there you
okay.
go.
Yeah,
Yeah.
yeah, yeah, yeah,
Yeah,
yeah.
and I also saw that you have videos with some microorganisms that have really cool features that I don't know if how common they are. For example, there was one that could switch from photosynthesis, like regular photosynthesis, to producing oil. Is that correct? Like, do you have some that you find are really cool and maybe a lot of
Mhm.
people don't know of, that this skill even exists in the world?
Oh, all of them, all of my subjects are like that. So it's just like they're super cool and nobody knows about them. Um, well, let me think. And what microorganism that I can give you an example of? Um, well, tardigrades, they're also called water bears. They're quite popular, I think, but they can produce these cysts, or they're also called tons. So basically, they roll into a ball and they just wait, when there are like stressful environmental conditions. They just wait and they can just survive probably anything, and you know they can just come out of the ton, and they can just be active again after probably hundreds of years. I think the longest scientific research on them was 30 something years from Japan. So they came back to life after 30 years of being frozen. But there are some reports of them from somebody from I think from Germany. I'm not sure. They put some moss into the water, and this moss was in the museum, storage for 100 years, and they found some tardigrade in it. And what else? Well, this is like a really broad topic, and I find everything super interesting. So it's really hard to just choose one. Oh, well, I can talk about one of my favorite ones. This is called Lacrimaria olor, and we made a really good episode about them. So this is just like a single cell organism, but it can just extends its neck 10 times of its body size. So you have this, 0.1 millimeter uh, microbe, and all of a sudden it's becoming like 1 millimeter, just extending its neck out to hunt. And they are gorgeous predators, and they feed on other microorganisms, and they can just it's unbelievable to watch them under the microscope. I can maybe link the video so people can go check it. Okay, I'm just adding it to the chat. Gorgeous predators, yes, double pun. Can you guys hear me?
Yes, sorry. I had a phone
Okay.
call now that wouldn't stop calling you. So
Is that my
yeah,
professor? Tell her that
sorry.
I'm gonna call her.
Oh, that would be funny. No, I guess it was something else. Yeah. So I think I probably missed the last few seconds. Do you want me to pin something on top? Oh, you might. Do you want me to put it
Uh,
on top?
uh, no, not really, because it's just like about the microbe that I was talking, and I think people that are listening now, they can just go check it, but it doesn't need to be on top of anything, I think.
And Dr. Shah, welcome and hi. Sorry that I've been taking over here a little bit. Did you want to ask a question, Dr. Shah?
Hi, Katerina, and our guest, James.
Hello.
Yeah, hopefully that you have a very nice Wednesday, both of you. Yeah, that was very fascinating topic, and I'm just listening because I'm learning. And when it's the time for asking, I'm going and asking my question.
Cool.
Yeah, anytime is good. So whenever you have questions, please, just let us know, and we'll go from there. Um, and one question in the chat I saw is that Sabil, he read your book, and it's a really great book, and is asking when you will be writing the next one, or are you currently writing another one?
Yes, I have a couple of book projects. They're gonna be, I think, published in 2023, late 2023. But I want to make something with better print quality, which increases the cost of the book. So I'm not sure how it's going to go yet, but I want something really high quality, and it's also like scientifically correct and everything. So it's just taking a bit longer, but I am working on it. Yes,
So
and
that's
thank you for
exciting.
reading my book. Yeah, I
Will there
couldn't
be
catch the name, but yes, thank you so much.
and will there be probably this pretty images, that's why you want the
Yes,
quality so
yes,
high, so the
yes,
images will
yes.
reflect the.
Yeah, because my first book, it also, has a lot of photos, but it was more like um, how can I say it? Like it's so we, it wouldn't be possible to print it on, gorgeous paper and just, because it would be making it like 5 times more expensive than it is now. So, but I am trying to prepare something that would be, financially accessible for anyone, but also has the best printing quality. So it just, yeah, we are working on it.
It's so impressive how many different projects you have going on, and you even consult for companies, right?
Mhm,
Because everyone is impressed with your microscopic skills. Um,
mhm.
so you even developed your own microscope. Did it help that you have some engineering backgrounds, and how did
Uh,
you manage to do that? Because I know in Clubhouse are quite probably the highest percentage of people that want to like have these different type of careers. So I think it's also really
uh-huh.
interesting how you managed to do that, like to have these different paths of careers and be successful at them.
Uh, well, I'm probably not the right person to give any advices because I'm not goal oriented, but I am more like passion oriented. So I just do things that makes me happy, and then we have a product, or then we have a book, then we have a paper. So I don't know, it's really strange and it feels unreal sometimes. But for the production of these microscopes, uh, well, it was quite straightforward because, like I knew what we need for a microscope, and so this microscope is based on the first microscope I got, and this is to introduce people to microscopy. So this is wanted to make the you know, cheapest proper microscope that somebody just can buy it and just start doing the things that I'm doing. So I sent a couple of emails to some factories in China, and I asked them about their products, and then we created something, and it's uh, yeah, but this is I am partnering with this. So this is not just my work. So there are so many people working behind this, and like our microcosmos team, and we are just, we are also working to create more products. So we are trying to make sampling, a bit easier for people, and we are creating some, like sampling accessories, like sampling sticks, or just stuff that would come handy if you go out to collect some water samples for your microscope.
Yeah, that's so interesting. And you keep mentioning mostly you were driven by passion. And do you think that you kind of could preserve your, like, could keep your passion pristine because you didn't go to like a traditional school for this? Why I'm saying this is, I did my undergrad in biology in Germany. I
Mhm,
always loved this type of, you know, uh, different types of animals
Mhm,
a
mhm,
lot. But when I then studied, I had to go out, collect samples, and then know all these families, and do this in this, looking it up in the book, and do it in a very specific manner. And I feel like always when I had to go through this classic education, the same with music, it kind of ruined making music myself for
mhm.
me, going through a classic music. It kind of ruined it for me. Like, do you think that is true? Like, what do you think about going through a classic education? Does it keep your passion pristine, or do you get like fed up
Mhm,
from
mhm,
the teachers and professors telling you
mhm,
exactly
mhm.
how to do it? And like, did it keep it alive, basically, that passion?
Well, um, it would be really hard to, say something super solid about it, but um, everybody's experiences, you know, could be different from each other. But I will never work in academia because of this reason. Because, uh, if you work in a lab, if you do the science that you have to do to get papers or get funding or get, whatever, then it's something that is not you anymore. So I think it's really important uh, to keep the. I'm reading Annie's comment now. Just yes, great. So um, sorry, just a second. Yeah, so I don't want to lose myself within competition or trying to find funding for my research or trying to get something accepted. I think it would frustrate me, and I think because of that reason I wouldn't be able to do that. So I would be choosing to do just the microscopy in my living room without getting any scientific recognition, without getting any public appearance, nothing. So I would just be doing microscopy. I would be choosing that over publishing papers and getting getting titles or just getting fundings, or it's not my cup of tea. It would kill everything, I think, about my about my excitement. uh, yeah, but I so there are some courses, there are some professors that teaching stuff in the field that I'm interested in. It's absolutely amazing to just join the lectures. I am even sometimes getting these courses from their websites that you can just get the courses online just without paying or something. You can just attend. So I'm just doing that quite often, and I love it. So I love learning more, but I wouldn't like learning more about stuff to just pass an exam, if that makes sense. you know
Yeah, that totally makes sense, and I agree. I agree with you, and congratulations to go that way. Was it kind of scary in between, or was it something you didn't really think about? Like,
no, I
something.
really didn't think about it. It's just like well, I didn't have anything to lose, so it wasn't even a decision. It was just the way that things happened, and it really wasn't something that I brood over and try to make the right decision or something. It didn't happen like that. It was just like, oh, I like doing this, I keep doing this, and then it becomes something, and then it become my only thing, which, you know, it's really hard to explain. And it's I often get these questions because people think that like I was trying to have a career. I was trying to, produce something. It wasn't like that. It was just a random guy just doing things that he likes. And, that's really everything. And then it become, quote unquote, my career, and it become my full-time job. It become something, other people are proud for me, proud of, and yeah, it's really strange. I cannot call myself lucky, because I think I did this, in a way that, I put a lot of effort into it. But so it's not like stars aligned and I become who I am now, but like it took a lot of effort, but it didn't felt like I'm putting any effort into it, because it was just pure passion, pure love for me. And yeah, it didn't feel bad, that I stayed awake for 3 days, just, trying to find something under the microscope. It, yeah.
Yeah, that's so wonderful to hear. And again, congratulations. And the work you do and this YouTube channel, it has so many interesting facts. I think it's also good for the science, like for the image of science, you
Mhm.
contributing really significantly to it, to have something genuine and interesting and beautiful at the same time. I think that's really important, especially nowadays. So did this whole COVID and everything like changed anything? Like, did you
Oh, I didn't
get
even
sort of
realize there was there
didn't?
was a pandemic going on, because I am super asocial, and just I'm in a country that I don't speak the native language, and I'm just living here, just doing microscopy. And when the pandemic happened, it was like, oh well, it changes nothing in my life. And like everything stayed the same. And I even joke about it. I was saying like, yeah, you know I was prepared for this for all my life, so it was absolutely okay to just stay at home as usual and do microscopy. And I have like 2 cats, and I love them so much. And when I leave the house, I miss them. So I don't leave the house that often, except for sampling and just, you know, doing like those short things. Uh, oh, I keep thinking that I dropped out from the call, but I didn't
Oh
really.
no, you're fine.
I
I just got another phone call. Amish is trying to speak. Amish, I'm trying to bring you up. Uh, yeah, ask away, Amish. Thank you for coming.
Thank you. Thank you, Katrina.
Hello.
Thanks, James, for the. So I have a personal question. Like,
Hmm.
um, since you spend so much time looking into the microscope, and
Hmm.
I think you also mentioned you're asocial. How has your perspective of this macro world changed, or how do you see this world differently than us?
Hmm.
Because there are so many microscopic things which we take for granted, or we don't even notice, and we
Yeah.
might be living our daily life at an edge, at risk,
Yeah.
or at maybe a next scientific breakthrough, but we are just chilling away our life. So how do you see these things? I'm quite
Uh,
fascinated by knowing you for.
yeah, I think like microscopy is a humbling thing because of many reasons. But it just when you look through a microscope, you see all these things. They are, and some of them are not single cell organisms. They're literally animals. They have like simple brains. They have, muscles. They have, eyes and stuff. They are just living their lives, and they are trying to avoid any kind of stressful conditions, and they are trying to feed themselves, and they are trying to reproduce. They are just trying to get the best out of their tiny lives. And so seeing this hours and hours every day, it just it gave me this understanding that I am part of this wonderful thing. I'm part of life. So it makes my individual existence in this planet is really small, but also it makes me really important, but in a way that like I don't feel like superior to these organisms, but I just feel that I have so much in common with them. So it makes me just appreciate life. It just I maybe that's one of the reasons that I keep bringing up the passion topic, because it just nothing else matters. It's my career, or like how much money I make, or they don't make sense if I'm not happy and just enjoying this short life. It's one day, um, maybe maybe not in 10 years, or maybe not in 100 years, but we humans, we're just gonna disappear. We're gonna go extinct, and but the life will continue. And I think that's really, really good, because we are going to be the part that that created that life in the future. So it just makes me feel like I'm part of something wonderful, and makes me so happy.
Awesome, awesome, James.
Yeah, thank you for the
There are,
question. Yeah.
just sharing this insight. It's quite common to have similar feelings among people who actually use a telescope and look into the
Hmm.
macrocosms day in, day in, out, day
Yeah.
out. Sometimes
Mm.
sometimes I feel there's a truth which is very similar if you go macro and go micro.
Yeah. Yeah.
Any-
Yeah. Yeah. Yeah.
Thank you, James.
Thank you for your questions. uh okay, somebody is
Uh,
asking,
oh, yeah, go ahead.
do you believe someone is using microscope to see humans? No, I don't think so. um no, I think it well, as a person who is using the microscope all the time, I think humans are not that interesting under the microscope. I think, yeah, I think the whole universe would look better. Uh,
Yeah, I agree. Ashish, you joined the stage. Welcome. Did you have a question? Please go ahead.
Yeah, James, I missed your entire talk, but I love your philosophy of life. Uh, in
thank you so
academia,
much.
in academia, we seek what we know, and those that sometimes miss out on academia, what they called idiot savants in the past, are actually the true
Hmm.
people, the true seers. And I love a couple of things that you said. First of all, that you will never join academia. Understand it completely. Truth is found in on your own path, as Jisnu Krishnamurti would say. And
Hmm.
the second thing is that you have to live in your passion, and I really respect that, because that's what life is about. We all need to earn money a little bit so you can feed your cat,
Hmm.
apparently. And
Hmm.
it's cool that if you don't like people, you can stay at home and do this awesome work looking at water bears and all kinds of other beautiful structures in life. That is just amazing. And I just want you to laud you and praise you that you should keep on doing what you're doing, and you'll do that anyway. So
Thank you so
just
much.
a
Yeah,
big thumbs up for you. And
thank you so much.
there's so many things I could ask you, but there's a room full of people, and I know how to find you. So take care, James. Have a good
Thank
one.
you so much. Thank you so much. It was lovely hearing this.
Yeah, I can only agree. Like, it's like in a little bit like um a fairy tale, James, because you do this amazing work and then it's your passion, and also the philosophy of life you have is very mindful and yeah, what life is about. So yeah, it's such a pleasure um, seeing your work and then also speaking with you. It makes the work even more impressive, and you know how it is, like when you're actually also impressed by the person, then you like the work even more, and it's more interesting. um, so I think Annie asked a question: is there any microscopy of your own gut bacteria, like did you ever try it, or can it not survive conditions for studying it? I don't know if it,
No, I
if
didn't
it's
check my own gut bacteria, but there's a lot of research, I think, on human microbiome. Let me read the comment. Can it not survive in condition for studying? Oh no, we can create these conditions under the microscope, so it would be maybe not under my microscope because I don't have that much of how can I say patience, I think, for that. But we can create these conditions to study them under the microscope and do research on human microbiome. But yeah, but also they recently discovered that, as you said, they're producing neurotransmitters. So our gut microbiome is affecting our mood, which is something fascinating. And that's only some of the things that we know so far, because this topic is just becoming more popular over the last decade, I think. Yeah. Oh, I saw the comment, but then it disappeared.
um yeah, I agree that there is a lot of gut bacteria research currently. Oh, Stabil, I see the comment now that you mentioned. Stabil is asking, I want to try to make a center Coriolis sample. Do you have any tips on how to make, how to look at them?
Add a couple of drops of milk into your sample.
Oh, milk.
And in 2 days,
Oh, wow,
like
interesting.
high-fat milk, because the nutrients in the milk, it just boosts the bacterial reproduction, and then things that eat bacteria, they just grow, and then stentor feed on them. So in a couple of days, you would have like thousands of stentors right under the water surface, and you can see them with a magnifying glass even. Are the microscopic creatures you're studying larger or smaller than gut bacteria? Well, some of the things that I study, they are larger than the gut bacteria, I think. But so let's say I have no idea about the average size of, because there are so many genera of bacteria and archaea living inside us. And so let's say there are like 5 microns in length. So some of the things that I'm studying there are around 100 microns, but I can still see the gut bacteria quite beautifully under the microscope. if I wanted to.
Yeah, I saw in your videos that you have like a big variety on the magnification factor you use, and that kind of
Hmm.
reflects on, you know, the different sizes of
Hmm.
the. Oh, I thought it was so interesting to see that bacteria also use tiny, tiny microscopic minerals to figure
Hmm.
out if they are upwards or downwards, like where up and down is, and
Hmm. Hmm.
also the tiny minerals. I don't know if anyone is interested. Check it out. This, I forgot which one of the videos it was exactly, where you see the
Hmm.
branch motion um, effect on these tiny minerals. That
Hmm. Hmm.
was that is so interesting. So all this knowledge that you ex- where, which books can you recommend? Some books, or where do you learn all these different facts that are so interesting?
Well, I think it's the hardest and the, you know, most, I think, enjoyable part of my work. So I need to dive deep into these references. Um, so these minerals and stuff, they are rather common knowledge in microscopy, in like microbiology, because there are a lot of eukaryotic organisms that produce these kind of crystals as their um, metabolism's byproducts, and we are not sure if they're using them as like a nutrient source or they're just like pure waste. But it's common. So the things that, you know, interesting and hard to find is that these old and rare species and stuff. And so to find the information for these things, I am constantly, going through these, I don't know, like 150 years old papers, that's written in German, in Russian, in French. So I don't speak any of these languages, but I have a good understanding of really weird scientific words in any of these languages. So it's so weird. I obviously I use Google Translate, and I ask to, like random people on my social media to help me with some, tricky translations and stuff. But it just it's like you just need to go down the rabbit hole. I think that's how you learn the most, effectively. So it just keep following the references, I mean the references as like, you're reading a paper and there's like in parentheses it says like, I don't know, Weiss, for example. If they gonna reference my paper, it's gonna be Weiss, 2022, and then you go the back of the paper and then you find the whole name of the paper, and then you find that one. So what I'm doing is I'm doing that in a like um, papers from, you know 1850s or like early 20th century and stuff. So it's really hard to find those papers, but it's so much fun to look for them, and you learn so much from this thing.
Yeah, that's really interesting that you go down these and holes, and that you then manage to find these papers. I agree, it's really hard. I sometimes tried it too, and then sometimes you have to go to an actual physical library. I don't know if that's still the case, but
Yeah, we did that a couple of times, and they well, you don't have to go to the library, but you contact the library, so they send you a scan of it. And yeah, and social media helps a lot because I was trying to find something from 1967, which is kind of new, not that old, you know. Um, but this was like it wasn't an article; it was just notes from a symposium, and it was in Russian, and it was from Soviet era, and we couldn't find it. And then I wrote something about it on my Instagram, and someone like I know online just sent a message to me and said, "Hey, I'm working in the old office of that guy that you're looking for, so I can just go to the library and just find that paper for you." So yeah, she went down to the library and she just came up with this like 2 sentences, literally 2 sentences thing that we were looking for I don't know, for weeks. Yeah.
That's wonderful. I'm so glad. Yeah, I agree. People on social media, there's like a lot of downsides, but there's definitely a lot of upsides where you make like connections that you would never do, and and people are helpful. That's
Yeah.
really interesting. And I know we've going on for an hour. Jamie just joined the stage. Is it okay if he asks one more question and then we go?
Yeah, I have time, no worries.
Okay. Oh, perfect. Thank you.
Hmm.
Jamie, do you want to go ahead and ask a question? Can you hear us, Jamie? Or
Um,
maybe he can't hear us right
in
now. Maybe he has the phone call issue that we had today.
yeah, my professor is calling
Still
them
calling.
now. Yeah.
Let me check. I don't.
These
Oh, yeah.
do you
Go ahead.
study prokaryotes and eukaryotes, and what's the difference in how you study them, if
Oh,
any?
can you hear me now?
Yes.
Hi. Yeah,
Hi.
we
Oh
can.
yeah. Sorry about that. I was locked into mute. Mute anyway. Sorry about that. James, thank you very much for that incredible talk. Your passion and persistence is actually pretty inspiring. Thank you very
Thank you so
much.
much. Thank you. Thank you.
And my question is this: Can what are you actually going to be working on next? And will we be hearing from you in the near future?
So at the moment, I have, maybe I shouldn't say the names at the moment, but I have, let me think, I have, I think, 6 paper Word data, and so I will be publishing them in, I think, a couple of months. Like, and they are super rare ciliates, which I'm quite excited about. And these are the things that I've been looking for years, and like I spent thousands of hours just doing microscopy and searching for them, like keeping my eyes open for them. But uh, yeah, as the tricky part is getting the genomic data for them after you found them. So we got that data, and we're gonna be ready to publish super soon. And probably I'm just going to be talking to my best friend. Also, she's my professor. And so I'm just going to be talking to her after this talk about our next paper, because I finished drafting it last night and I need to send it to her. Yeah, so these are rare microscopic organisms. Yeah.
That is incredibly exciting. I'm looking forward to hearing more about that. Thank you very much.
Thank you so much. Thank you.
Somebody asked, and Ridi asked where she can. I know I posted Ridi some resources on top of the chat, with the book, where you can find the book, the paper, and the YouTube channel. But do you have maybe a website link where everything is like, all the links are, or maybe a link tree URL or something?
From my side,
Yeah.
I suck at the self promotion. Really, I don't have a website. I need to get a website, and it just, yeah, I cannot just put everything in one place. But I should have some link tree. But I'm not sure if it has everything. Published papers. Yeah, I think on top of the chat here.
Yeah,
Let me
they're
share.
already
Yeah.
there. We cannot put the links back again, like a Clubhouse.
Uh-huh.
Whenever you share the exact same thing again, it blocks the share. I guess they want to prevent that
Oh,
people
yeah,
spam a lot,
yeah,
like
yeah.
because some people just come in and promote, I don't know, their crypto thing, or you know, the moderators have to constantly delete, so they kind of block that you keep putting in the same information in the
Yeah,
chat, I guess.
yeah, makes sense.
Yeah, I agree.
Uh,
Um, well, and also, yeah, please check out on top. There is the link to the book, the link to the paper, the link to the YouTube channel, and even the store that James and his colleagues have set up, where you can buy things, even microscopes, which is really impressive.
we don't have them at the moment, I think,
Oh,
because
you don't have them? Okay.
the thing is, like, we restock them and then we ran out of them in like 2 days. But I think within a week they're going to be available again, because we get a big shipment. So people can subscribe to the email notification, and they would get the notification when we restock them.
That's really cool that you sell out so fast. It's so impressive that, you know, with this scientific channel, and you know that people are so excited about this, that you sell out immediately. It's really, it's very positive. It makes me feel
Yeah,
more
yeah.
positive about
It
this.
it makes me so happy. It just
Yeah.
like you're touching so many people's life, right? Because it's I cannot express how much I love microscopy. So if somebody, just gets a microscope because of us and just, start doing the microscopy and just have this, just a bit of my passion, then it means so much to me. It's unbelievable. I think it makes me
Yeah,
so happy. Yeah.
I can relate. I'm so happy when people say that our club here kind of changed their perspective on
Mhm,
science. A few people wrote me they went back to school again to study
mhm,
again as they listen.
mhm.
Like, I agree, it makes me also very happy. So I
Yeah,
can
yeah,
relate, and it's amazing the
yeah.
work you do. It's And then Siva asks, last question, if time supports, some lecture on relation between microscopic plants versus microscopic species or animals, I guess. Like, is it more difficult, easier? Um, you know.
Um, um, uh, I'm not sure. Is this a question?
Yeah, it was in the chat, the
Or
question. If you click on the chat, that's the last comment
mhm,
there.
some lecture on relation between microscopic plants versus microscopic species. Uh, Microscopic plants versus microscopic species. Well, I'm not sure if I'm getting it correctly. Then,
I
Siva,
know you
maybe
had
maybe
the,
maybe maybe you can explain it a bit.
well,
Uh,
yeah, I don't know if I get it specifically too, but I can remember that you had a video that kind of showed like a symbiosis of mini algae with like a pretty big microscopic, quite
mhm,
common species that kind of protects the algae, but then the algae give them some sugar. Like, are there many
mhm.
examples like this? I think maybe that's the question pointing at.
Yeah, the symbiosis, like it's called the endosymbiosis. So there are many examples of it in the microscopic world, but these algae are still single cell eukaryotes, so um, they are not plants as we know. Um, so what I do is like I specialize on ciliates, but also I work on protists, which is basically everything microscope, which is everything that is not animal, not plant, and not higher fungi. So it just, it's it's commonly called the dumb taxon, because if you cannot fit something into these, you know, these definition, then you put them under the protist. So we have a lot of, photosynthetic protists. We have protists that, like actively need to acquire energy by consuming other organisms. And yeah, so it's a really wide topic, and there are so many examples in, like amongst protists, that this endosymbiotic relationship of, photosynthetic organism and one host that is not able to do photosynthesis, but the host always, like if it's a species specific relationship, which is also common. So there are some species that you can only find them in nature with their endosymbionts with the algae, so you cannot find them without the algae ever. And so from generation to generation, when they divide, they also divide the content of these endosymbionts. So there's, let's say there's one organism with hundreds of this single cell algae living inside it. When it divides into two, it just, it divides into two, and the two new cells that are called the daughter cells, they now have 50 algae each. Now it's not correct in a numerical sense, but it's just they share the algae as well. And then, this goes on from generation to generation. So these relationships are, most of the time, they're permanent, but they can be also temporary. Some organisms just can, acquire algae from the environment, and instead of digesting it, they can just keep them inside, and they share the resources the algae produces from algae produced from the photosynthesis. And there are very interesting examples of this. For example, there are some microorganisms that can that have these algae inside, and they protect algae from viruses. And it's so interesting because the virus cannot infect the algae. And yes, that's also a thing that, some microscopic organisms get infections as well, and viral infections as well. So it just, the host is just providing like a immune system to, its endosymbionts.
Oh wow, that's so interesting. That it's providing the immune system. That's fascinating. Thank you for that question also.
I have a question, if I
Yes.
may ask. So, James, what about the quantitative information? Because at the beginning you just mentioned about producing data, and I was just wondering to ask you about transcriptomes and those kind of data,
Mhm.
first of all. And when you just compare all of those data, and you explain about the colonies. So, what was the role of the invasive species in comparison with the symbiosis in those colonies? Did you find any further information around that?
um, I'm not sure about the question though. Colonies of what?
Let's be back to the first part, which is the
Okay.
data from the transcriptomes. So, did you, I mean,
Uh,
during
I
your
didn't
work?
mention the
Yeah,
transcriptomes.
I'm
I think it was
asking you,
me.
is that possible that
Okay. Um, Not really, because the microbes that I'm working on, they are super rare, and we are just focusing on the, like a partial, um, just part of their genome. So we are not doing anything, like advanced in genomics, but we are just acquiring the 18S data, and then we are creating the phylogenetic tree based on the 18S data. And in most of the cases, it's just, like our microbes, they don't have any genomic data in the genus or in the family. It's just like, it's really, really tricky to do anything more advanced than just creating phylogenetic trees, based on the 18S sequencing.
I see. It means that it's never go to the mRNA level or kind
No.
of hybridization or those kind of things. And the second part that was about the invasive species. Did you find any invasive species? And what is your, comment about those species?
I've never found an invasive species, I think, but it's but I'm not sure if there's a term, in microbes especially, that I don't think a microbe can be invasive. You're not asking for like causing infections or something, right? This is just like,
No, in between
yeah.
in comparison in between other species. Actually, that's a novel term that they are using, and I was just wondering maybe you have further information.
With microbes are like, they are cosmopolitan, so you can find them everywhere, and it's not. I don't think any microbe is called invasive before, so it just because it's common to find them everywhere. It's just they distributed everywhere, and if it's their habitat, you can find them. And if it's not their habitat, if they, for example, if a freshwater microbe ends up in, like in a marine habitat, it cannot just live there and it just dies. But so if a microbe prefers to live in, this anoxic sediment of freshwater habitats, there's a chance you're going to find this microbe everywhere. There is this anoxic sediment with the same properties or with the preferred properties, regardless of the, geography. You can find them in China, you can find them in Brazil, you can find them, in the US. So it's, um, yeah, they are not invasive, but they're just, natural, I think. Invasive species, I think they appear more when you become, when you go higher in the size, and then yeah, that's the thing, I think.
Yeah, thank you. Annie had a last question. If you still have a few minutes, if there's any viral symbiotes, like the symbiosis you mentioned before, if there's
Hmm.
something like that with viruses,
Hmm.
and yeah, and is there any of those classified as such that don't have endobiomes? Thank you.
I'm not sure if it would be possible to have viral in the symbionts, but you can have, although like it's not my expertise, but you can have some, viruses as dormant in your cells, but that doesn't mean they're endosymbiotic. Endosymbiosis is um, more like, the host is providing something for the endosymbiont, and endosymbiont is providing something for the host. But since viruses, they get activated within the cells, I don't think they can have this kind of relationships. But as I
Yeah, I
said, it's viruses are not my expertise at all. So I've never seen one under the microscope. Um,
yeah, and
well,
the only thing I can think of, we had the guest speaker here that in the mangrove, where mangroves grow, they found these humongous bacteria. They are really, really
mhm.
big, and they have kind of organelle compartmentalization,
Hmm.
and I think the viruses that live with them basically give them the ability to form these organelles, partially at least. I think that's the only, but it's not really symbiotic, but it's maybe goes a
Hmm.
little bit in that direction. That's the only example I can think of. I have to find the
Um,
paper. I don't have it right now, but yeah, if
paper, and
somebody is, then I
yeah, I'm
can.
familiar with the research team behind that discovery as well. Yeah, but I don't have any remarks on the endosymbiotic viruses, I think. Yeah.
Yeah,
Research
and then
discussion.
Annie also asked, how would you, like, what would you need to be able to visualize a virus?
No, why you cannot see viruses with a light microscope because they are smaller. Well, they're giant viruses, but they're still, I think, 400 nanometers or 500 nanometers, which is super small still. So it's they're smaller than the wavelength of the light, so you cannot visualize them with a light microscope. And you need an electron microscope, either like scanning electron microscope or transmitted electron microscope, to be able to create a picture of them. But it's impossible to do that with a light microscope.
Yeah, and then you cannot have this pretty video because
No, you can.
electron microscopy is not fresh. Like,
Nobody can make a video of a virus yet, I think. Yeah, I'm not sure about the advancements in electron microscopy and stuff, but no, you can just take photos because you need to fix your subject on like a metal plate, and you need to cover it with I don't know, in most of the cases with gold, I think. It's just you need to fix your thing, and then you need to shoot electrons on it and just create a picture from the reflected electrons, I think. So you can only get a video of it. You can only get a photo of it, or a very still one frame video of it. Yeah.
yep, yeah, I agree. And yeah, thanks for all these interesting questions. And James, thank you so much for answering all of these questions. And it was such an honor having you here. And we will follow your research work, video making.
Mhm.
At least I will follow it. And it made it even more interesting. And yeah,
Yeah. Thank you so much
more
for
than
having me, and it was great talking to you guys. And yeah, I hope it was fun to listen.
yeah, I had a lot of fun. I hope you had fun. And yeah, I hope you'll come back one day, maybe next year or so when you
Of
published your
course. Yeah.
papers. That would
Yeah.
be amazing. And
Yeah.
yeah, everyone follow this YouTube channel. Is amazing. It's really interesting. And yeah, stay tuned for the microscopes that will be available soon. And
Thank you
then you
so
can
much.
discover microbes, maybe. Okay, thank
Yeah,
you,
it's
James.
possible.
It was
Yeah.
a
Thank
pleasure.
you. Thank
Thank
you.
you.
Bye bye. Bye bye. Thank you so much.
Bye, James. And oh yeah, just a quick announcement. Follow the club if you like discussions like this. Tomorrow we'll have Dr. Oliver talking about how he managed to use aluminium nanoparticles to produce hydrogen out of regular water. You know, hydrogen is important for like different type of fuel, and it's quite expensive and not easy to do, and he's working on that type of research. And then we'll have a room next week Monday again with Dr. Ed Boyden from MIT. If you never heard of him, check him out. He's one of the most impressive and also important technology development for neuroscience tools. He did like major technology development that we all use. So he will talk about this work, tools that image and control the brain. He's really nice and really impressive and a great person to talk to. And it will be a similar style. He did so much important work. You cannot really just have one paper up here. That would be way too limiting. So I will do the same thing. I'll post his website and different papers in the chat, and then we'll have a discussion where he describes his work and the impact and how he came to do this work. So thank you so much, everyone. Maybe hear you again tomorrow. Will be an interesting room, and yeah, enjoy the rest of your day, morning, wherever you are. And thank you, Dr. Shah, for
Thank you,
moderating.
Katerina, for holding such a wonderful room. The same as always, and saying bye to everyone.
Thank you. Okay, close the room in 3, 2, 1. Bye, everyone.